Disaccharides. Disaccharides are composed of two sugar units. It is a carbohydrate in which two monosaccharides are bonded together. In disaccharide formation, one of the monosaccharide reacts as hemiacetal and the other functions as an alcohol. The bond that links the two monosaccharides together is called glycosidic linkage. A glycosidic linkage is the bond in a disaccharide that results from the reaction between the hemiacetal carbon that contains the hydroxyl group of one monosaccharide and another hydroxyl group on the other monosaccharide. It is always a carbon to oxygen to carbon bond in a disaccharide. One of the disaccharide is maltose. Maltose is often called malt sugar. It is produced whenever the polysaccharide starch breaks down. This happens in plants when the seeds germinate, and in human beings during starch digestion. It is a common ingredient in baby foods and is found in malted milk. Structurally, maltose is made up of two D-glucose units, one of which must be in an alpha-D-glucose. The glycosidic linkage between the two glucose units is called an alpha-1,4 linkage. The two hydroxyl groups that form the linkage are attached to carbon 1 of the first glucose unit and to carbon 4 of the second. Maltose is a reducing sugar because the glucose unit on the right has a hemiacetal carbon. This is in carbon number 1. The most important chemical reaction of maltose is that of hydrolysis. Hydrolysis of D-maltose, whether in the laboratory flask or in a living organism, produces two molecules of D-glucose. Acidic conditions or the enzyme maltase is needed for the hydrolysis to occur. Next, cellobiose. Cellobiose is produced as an intermediate in the hydrolysis of a polysaccharide cellulose. It differs from maltose in that one of the D-glucose unit, which functions as hemiacetal, must have a beta configuration. Cellobiose has beta 1,4 glycosidic linkage. Like maltose, cellobiose is also a reducing sugar. For the hydrolysis to occur, the enzyme cellobiase is needed to produce 2 units of D-glucose. As you can see on the picture, our glycosidic linkage is on the carbon number 1 of glucose and on the carbon number 4 of another glucose unit. Next is lactose. Lactose is made up of a beta-D-galactose and D-glucose unit that is joined together by beta-1,4-glycosidic linkage. Lactose is also a reducing sugar. It is the major sugar that is found in milk. Enzymes in mammalian mammary gland take glucose from the bloodstream and synthesize lactose. Lactose is an important ingredient in commercially produced infant formulas that are designed to stimulate mother's milk. Souring of the milk is caused by the conversion of lactose to lactic acid by bacteria in the milk. Pasteurization of milk is a quick heating process that kills most of the bacteria and retards the souring process. Thus, lactose can be hydrolyzed by acid or by the enzyme lactase to produce galactose and glucose units. Again, as we can see on the picture, the glycosidic linkage that occurs in lactose occurs in the carbon 1 of our galactose and carbon 4 of our glucose unit. The next one is sucrose. Sucrose is known as the common table sugar. It is the most abundant of all disaccharides and occurs throughout the plant kingdom. It is produced commercially from the juice of sugar cane and sugar beets. The two monosaccharide units present in the sucrose molecule are alpha-D-glucose and beta-D-fructose. Thus, the glycosidic linkage is alpha-beta-1,2 glycosidic linkage. The hydroxyl group on carbon 2 of the D-fructose, which is the hemiacetyl carbon, reacts with the hydroxyl group on carbon 1 of the D-glucose, which is the other hemiacetyl carbon. Sucrase, the enzyme, is needed to break the alpha-beta-1,2 linkage in sucrose. This will produce glucose unit and a fructose unit. This enzyme is present in the human body. Hence, sucrose is an easily digested substance. When sucrose is cooked with acid-containing foods such as fruits or berries, partial hydrolysis takes place, forming an invert sugar. Jams and jellies prepared in this manner are actually sweeter than the pure sucrose added to the original mixture because 1-to-1 mixture of glucose and fructose tastes sweeter than sucrose. Oligosaccharides. Oligosaccharides are carbohydrates that contain 3 to 10 monosaccharide units. They are also bonded together with the help of the glycoside linkage. There are two important oligosaccharides. We have the raffinose and stachyose. Raffinose is made up of galactose, glucose, and fructose. Stachyose is made up of galactose, glucose, fructose, and another unit of galactose. The blood type of a person is determined by the type of oligosaccharide that is attached to the person's red blood cells. One of the monosaccharide present in the oligosaccharide that is associated with blood type is 6-deoxy-L monosaccharide. Another oligosaccharide, we have solanine. This is a compound that is found in potato plant. Potatoes also produce a toxin in order to fight against the predator. Solanine is the potato's plant toxin. This trisaccharide is present in the solanine's structure. It involves two common monosaccharides, namely D-glucose and D-galactose. It also contains a rare monosaccharide which is named L-rhamnose. Polysaccharides. A polysaccharide is a polymer that contains many monosaccharide units. They are also bonded together by glycosidic linkage. Polysaccharides are often called glycans. Important parameters that will distinguish various polysaccharides or glycans together are the following. Number 1, the identity of the monosaccharide repeating unit in the polymer chain. The more abundant polysaccharides in nature, they contain only one type of monosaccharide repeating units. Such polysaccharides include starch, glycogen, cellulose, and chitin. They are examples of homopolysaccharides. A homopolysaccharide is a polysaccharide in which only one type of monosaccharide is present. Polysaccharide whose structures contain two or more types of monosaccharide includes hyaluronic acid, heparin, and these are heteropolysaccharide. When we talk about heteropolysaccharide, these are polysaccharides in which they contain more than one type of monosaccharide monomer. Number 2 is the length of the polymer chain. Polysaccharide chain can vary from less than 100 monomer up to 1,000,000 monomer units. Number 3, the type of glycosidic linkage between monomer units. As with disaccharides, there are several different types of glycosidic linkage. Number 4, the degree of branching of the polymer chain. The ability to form branch chain structure distinguishes polysaccharides from the other. So these are the proteins and nucleic acid. The first example of polysaccharide is a storage polysaccharide. This is a storage form for monosaccharides. They are also used as an energy source in cells. The most important storage polysaccharide are starch that is found in plant cells and glycogen that is found in animal cells. Starch is a homopolysaccharide that contains only glucose monosaccharide units. It has amylose and amylopectin. These are the two different polyglucose polysaccharide that can be isolated from starches. Iodine is often used to test for the presence of starch in a solution. A starch that is present in a solution will turn dark blue to black when the iodine is added. As starch is broken down through the acid or enzymatic hydrolysis, then the blue to black color will disappear. Glycogen, like the starch, is a polysaccharide containing glucose units only. The glycogen is ideal storage form for glucose. It is the glucose storage of polysaccharide in humans and in animals. That's why it is called animal starch. They are usually found in our liver cells and muscle cells. We have the term glycogenesis and glycogenolysis. From the word itself, genesis, it means to say glycogenesis is the formation of glycogen. Glycogenolysis, from the word itself, we have the lysis. This is the decomposition or the breakdown of glycogen. Structural polysaccharides. A structural polysaccharide is a polysaccharide that serves as a structural element in plant cell walls and animal exoskeleton. The two most important structural polysaccharides are the cellulose and chitin. They are both homopolysaccharides. Cellulose is the structural component of the plant cell wall. They are mostly the woody portions of the plant. Cotton is almost 100% pure cellulose since it is made up of 95% cellulose. Cellulose contains at about 5,000 glucose unit chains. Chitin is the second most abundant naturally occurring polysaccharide. Its function is to give rigidity to the exoskeleton of the crabs, lobster, shrimp, insect, and other arthropod. It also is being found in the cell walls of the fungi. Acidic polysaccharides. Acidic polysaccharide is made up of a disaccharide repeating unit in which one of the disaccharide component is an amino sugar and one of the other component has a negative charge. This is due to the sulfate group or a carboxyl group. Acidic polysaccharides are heteropolysaccharides. Two of the most well-known acidic polysaccharides include hyaluronic acid and heparin. The structure of hyaluronic acid contains alternating residues of N-acetyl beta-D-glucosamine and D-glucuronate. Highly viscous hyaluronic acid solutions serve as a lubricant in the fluids of joints, and they are also associated with the jelly-like consistency of the vitreous humor of the eye. Thus, the Greek word hyalos means glass. Hyaluronic acid solutions have a glass-like appearance. The next one is heparin. This is a small, highly sulfated polysaccharide with only 15 to 90 disaccharide residues per chain. It is a naturally occurring blood anticoagulant. It is also present in mast cells and is being released at the site of tissue injury. It prevents the formation of clots in the blood. Glycolipids and glycoproteins. A glycolipid is a lipid molecule that has one or more carbohydrate units covalently bonded to it, whereas a glycoprotein is a protein molecule that has one or more carbohydrate units covalently bonded to it. Glycolipids are called cerebrosides and gangliosides in the brain tissue, while glycoproteins are called immunoglobulin when they serve as the key component of the body's immune system in response to invading foreign materials. In glycolipid and glycoprotein associated with cell membrane, the lipid or the protein part of the glycolipid is incorporated in the cell membrane structure, and the carbohydrate part functions as a marker on the outer cell membrane cell face. That ends our carbohydrate. Thank you, and I hope you learned something today.